PhD in IRI. Then he joined IRI as a scientist. Then he became a principal scientist in 2010. In 2010, then later he moved to University of Arizona or University of California as a visiting fellow, and there his real success story started. So there he got a lot of papers in the reputed journals like Nature, PNAS, Trends in Plant Science, Genes and Development, and those are all very key papers actually in the field of ABA signaling, cold stress, and siRNA, microRNA. So without citing those papers, it is very difficult to write those papers in those fields actually. So those are the very key papers in those reputed journals. And he got many awards like ICR Jawaharlal Nehru Award and Boys Cause Fellowship and J.J. Sinha Gold Medal Award, several awards, and the list is endless actually. And he is a very key person for establishing this World Bank funded Nahib Cost project, and he is the person behind the success of the phenomic facility that you guys visited few days back. So without further delay, on behalf of everybody, I request sir to deliver the lecture. Thank you, sir.
Thank you, Dr. Naresh, for giving a nice introduction and also writing that write-up which was given to in the manual. So today I will be discussing with you on abiotic stress tolerance of crops. So my Sivani, you click on that one. It is not moving. So today I will be talking on abiotic stress tolerance. So in the environmental factor increases from 0 to say 100, so the growth start increasing at it reaches maximum at optimum level of growth factor, then after that it decreases. So the response of the genotype, these are some example, it may be a water, temperature, nutrient, light. So the response of the genotype is like this, like some genotype they may have higher growth or yield when the optimum factor is less quantity, whereas some other factor it may need even for the starting the growth it may need more amount of factor. So there is variation in this range, as well as there is a lot of variation in the range where the growth factor exceeds. So in these case of plant responses, the tolerant genotype may respond like this, and susceptible genotype may respond in this way. So these are some of the factors. So it may be water deficit, hypoxia, anoxia, temperature it may be high or low, nutrient also, like when it is deficient or if it is toxic, it's a problem. Light is also low and high. There are several other factors which are not required for the plant growth, and they can also cause stress to the plants. These factors are include ion toxicity, which are caused under salinity, alkalinity, or it is caused by the heavy metal. And the proton concentration in the soil is essential because often the nutrient availability or the growth of the root in the soil depends upon the soil pH. So each nutrient availability is determined by a certain range of pH. So if the pH is very high or it is very low, then also it affects the growth. Although the hydrogen ion concentration per se is not required for the plant growth, it produces a condition which is necessary for the nutrient uptake and the growth of the root in the medium. Air pollutants, it may be a gases or it may be just particle matters that are deposited on the leaf. Like especially if you go walk around in the campus, you can see a lot of particulate deposit on the leaf. So they also affect gas exchange process as well as light interception, and thus they affect the yield. And the UVB radiation, which was a major problem earlier, but now because of the ozone hole and other thing, now since the ozone hole is cured, so this problem is less. So in this, if you see the response of the plant, it is highly contrast to the response of the plant for the essential growth factor. So here you can have a resistant genotype. Whatever the stress is there, the plant will not bother. It will be resistant. It will grow irrespective of the concentration of the non-essential environmental factor. Whereas some of the tolerant genotype, it may tolerate to a certain extent, then after that you will see a sensitivity. And the susceptible genotype, the slope is larger when compared to the tolerant genotype. And also the critical tolerance level may vary between the tolerant and susceptible genotype. So we should understand this when we talk of stress tolerance. We cannot talk of tolerance to drought versus tolerant to other abiotic stresses. Often we think that we can generate a resistant plant, drought resistant plant or temperature resistant plant. That is not possible because when the stress is caused by an essential environment factor, you can have only the plant of a tolerant type. If it is non-essential factor, there is a possibility to develop a resistant plant.
Sir,
Yes.
how will you define that nutrient deficiency in excess and there is a ion toxicity? Both are not similar.
So in the nutrient, I put only the essential elements. So if it is caused by non-essential element, then it comes to the toxicity. If it is all the elements which are present in the soil is not essential.
Okay, so that is regarding the soil
Soil, yeah.
or it is regarding to
This
plants?
is with reference to the plant.
Okay.
If it is not required, like many elements are not required for the plant, then they are like coming under the toxicity stress. So in order to understand these responses, first we have to understand the mechanism of tolerance or the mechanism of susceptibility, and then we have to understand what are the genes that governs the mechanism of tolerance or susceptibility. Then only we will be able to edit that gene and improve the stress tolerance through genome editing. So this you all know. So the phenotype or the trait which you are interested in, that is either yield, stress tolerance, it is a product of genotype into environment. So when we measure this phenotype, we should measure very critically the environment, because that causes the phenotype which we see. But often we ignore the environmental component, and that's why there is a lot of difficulty in making progress in the stress tolerance research. So what are the different approaches for identification of the genes for stress tolerance or susceptibility? These are some of the approaches I have put. There may be many more. So the most important one is you have to precisely phenotype to identify the genotype or mutant which are tolerant or sensitive. That is the key. Once you have a germplasm which is clearly tolerant or it is sensitive, then you will be able to identify the genotype that causes, that is the gene or mutant that causes. There are several approaches. Often these approaches like have led to identification of many genes that are involved in the stress tolerance. Although recently we see a lot of these approaches, transcriptome, proteome, metabolome and ionome. These approaches have also generated a lot of information, but often they are limited, as you know that when you impose stress, you see large number of changes in any of the worms. So that's why it is very difficult to identify what is the causal factor. And also often the stress imposed in many of these studies, if you see, they are like uncomparable between the genotype, and that also complicates the inference from this. I will give some example for this. So when you impose stress, it is very important to measure the stress in the environment and measure the stress level in the plant, and then you see the stress response. These are three important factors for identifying the gene. Otherwise, if you just impose stress and if you identify a gene, you don't know what for the gene is working. Then when you want to deploy the gene in an environment or in a target population of environment where the genotype is going to be cultivated, that gene may or may not work. So that's why you have to measure all these three components. So this is the way you phenotype, you identify a gene that is going to be a candidate gene. So if you impose a stress under the field condition, here you see these are rice plants, they are subjected to drought stress, and they are grown for like the 7 days, no irrigation was there. You can see the cracks on the soil, but still you see there is very less obvious symptoms on the plant. So under this condition, if you screen genotype, you will find some are like tolerant and some are sensitive. So if you do like a transcriptome of this leaf and this leaf, you will definitely identify large number of genes, but you will not definitely identify the genes that is responsible for providing this kind of phenotype. Because when you do phenotyping under the natural field soil condition, the dehydration, cellular dehydration avoidance mechanism is the one that predominantly expresses in the soil environment, where the plant employs deep rooting system. So it takes up water from the deeper soil layer. So in this case, if you do a leaf transcriptome and identify, or you try to identify genes and you can make gene network, you can do many things, but you will not be able to identify the reason why the plant is tolerant. That's why it is important to understand what you are doing. And if you do phenotype in the pot, I am not saying that phenotyping in the pot is not important. It is very important to do phenotype under different conditions to identify genes that are responsible for different mechanism of stress tolerance. So if you do a phenotyping under the pot condition, you can see that within 3 days you face extreme stresses. In this you can find some genotype which are tolerant, some which are sensitive. So under this condition, if you identify, do any work and identify genes, this will be mainly responsible for the cellular dehydration tolerance genes. That is, even when the cell water relations are poor, the plant is able to survive. But in these conditions also, when you compare across the studies, like from one place to one place, if you conduct experiment there, you may see a difference. Like here, this is the pots are kept in open, so within 3 days they dry. But if you keep the pots in a greenhouse where the relative humidity is normally high, where we do experiment, the lab experiment mostly in greenhouses where the relative humidity is high. When the relative humidity is high, the vapor pressure deficit in the air is less. So the vapor pressure deficit is the one that drives the transpiration. So when you, the transpiration determines how much amount of water the plant is going to use besides the leaf area. So that's why there is, like within 3 days, these plants reached almost a drying stage but these plants are still alive. So here two factors are important. When you compare the genotype, you should see what is the leaf area produced by the genotype when we are doing a pot experiment. So if a genotype is producing large amount of leaf area, you cannot compare that genotype with the genotype which is producing less leaf area. But if you see the literature, you can find several examples wherein two plants are compared to transgenic versus wild type, where the transgenic plant is initially it is small. So when it is initially small, when you subject to stress, this plant is going to use less water, even if it is not tolerant. It will have a lot of moisture reserved in the pot, which can be used in the later stages. So that's why it is critical to see whether the leaf area is same when you compare the genotype and the vapor pressure deficit is similar. So that's why you have to do phenotyping under different conditions to identify genes for either the tolerance versus avoidance mechanism. You need both the mechanism to produce a better stress tolerance plant. Often if you see these two genotype here, this genotype, you see the leaves, they are almost fully turgid, and compared to these plants, they are almost experiencing the stress. But if we look closer, many of these panicle, they are white. That is, this genotype, although they perform better for the vegetative stage stress tolerance, that is, this genotype can maintain the leaf water status better, but the reproductive failure is more. That is, it is highly sensitive even for the mild amount of stress. So when you analyze the tolerance, you have to analyze the stage wise. That is, some genotype may be tolerant at seedling stage, or some genotype may have tolerance at vegetative stage, but for the reproductive stage, they may or may not be tolerant. So you have to identify genes that works at different tissue level and different growth stage level, so that you will have genes that are useful for the genome editing. So here some examples are given, like if you see in the literature, in many paper you find that we conducted the experiment and the soil moisture, either often it is not given, it will say 15 days water was withheld. So it doesn't say anything about the stress, only you know that the water was not given for 15 days. And some people give soil moisture content. So that is also not sufficient to understand whether stress was there or not there. Here I have given some example to illustrate that. So this is the plant's two genotypes, rice genotype, Nigena 22, which is a known drought tolerant genotype, and IR64, which is a drought sensitive genotype. These are grown in one experiment where it says the soil moisture content, it says it is about 25%. Still there is a lot of difference. This plant also experiences stress, that is relative water content. If it is about 90 or more than 90, there is no stress, but as it decreases below 70, it is a stress where the incipient plasmolysis takes place and the turgidity loss takes place. So here you can see both the genotype experiences stress, but this genotype performs better than this genotype. Another person is also reporting, he reported that the experiment was conducted, the soil moisture condition was only 20%, but still there is no difference. You expect that this difference is more, and the relative water content also should be, it will be still less when compared to this. So can anyone say why these differences are there? So this is very good. This is because of the different kind of soil. So if you use maybe sandy loam soil, the water available water will be ranging from maybe 7-8% to about 20%. If you use clay soil, maybe the available water start from 20% of the soil moisture to about 50% of the soil moisture. The soil, each soil particle, I think most of those who are from agriculture might have studied in their undergraduation. Each soil has different kind of soil particle, and the soil particle binds to the water. So depending upon the soil matrix, water binds to the soil particle, and the soil matrix potential varies. Matrix potential means the extent to which the water is bound to the soil. If it is strongly bound, even though the moisture is there, but it will not be able to reach the root, or the root will not be extract that soil moisture. So it is important to measure what is the kind of soil you are using, and if possible, if we can express the soil water status in terms of soil matrix potential, which is will be very useful for comparing the genotype or comparing the experiment. So this is a clay soil which has high water holding capacity. At the same time, the permanent wilting point of this clay soil is about maybe about 20-25%, whereas in case of sandy loam soil, even the field capacity will be about 20%. So which means the maximum available water here it is at 20, but here the minimum available water is 25. That's why we see a lot of difference in this one. So better when we do experiment, when we measure the express soil water status in terms of matrix potential. So these are some examples of how to you measure the matrix potential versus the water content. And often in many papers, I also find the soil moisture content is like expression of soil moisture content is also not correctly expressed. That also is it. Remember, it is expressed per unit dry weight. Normally, if you want to know the water content, see how will you calculate water content? So in a fruit, if you want to say express water content, how normally we express? Fresh weight minus dry weight divided by? So total weight, that's what normally we do. Fresh weight minus dry weight divided by the total weight. But in case of soil, it is fresh weight minus dry weight divided by dry weight. So to compare among different soil, we use that expression. So that is also will make lot of difference if we use the weight of the soil per se. It will be different because of the particle nature of clay soil will be different, and the particle nature of sandy loam soil will be different, so the weight will be different. That's why we use on dry weight basis. So this most of you might be knowing. So when you give irrigation, like when you study some paper, they say the control plants were given 20 irrigation, and stressed plants were given 5 irrigation, and still the stressed plant performed better. So in some cases, the plants may not require too much water. Say if you take wheat, we give only 5 irrigation. So it also important like what crop you are dealing with. So maybe control plant you irrigated daily so much, it experienced already stress, hypoxia or anoxia stress. That's why control plant is not performing better. But the other stress plant you gave water which is just sufficient to maintain the water in the field capacity or above 70% of the field capacity. That's why the plants performed under those soil moisture conditions. So is it understand the soil characteristic and what is the field capacity of the soil and what is the permanent wilting capacity, and based on that you have to maintain irrigation to provide a proper stress. And it is also important to understand how to express the results when you get in the stress versus control plant, especially when we are working with drought stress. So here it is another example is there. Here they studied the proline accumulation. Proline, you know, most of you might have studied. So what is proline? So it is an amino acid, essential amino acid, but it is also an osmolyte. So it accumulates under many stress conditions, and often we measure proline accumulation, and we say if it is accumulating high proline, it may have important role in stress protection or osmotic adjustment. So here the person measured the proline content, and he also studied the expression profiling of the genes involved in the proline biosynthesis, say delta 5 pyrrolin C carboxylase expression he studied. And in the proline content, he did not see any difference in the between the genotypes. So both the genotypes accumulated. This is under drought stress. So about 17-18 microgram per gram fresh weight of the leaf. But the expression of genes for proline biosynthesis was higher in Nagina 22, and there was no difference in the genes that is coding for the proline catabolism. So there was a difficulty in explaining the results. So what could be the possibilities? Why it happens? Assume that the gene is expressed and the expression is proportional to the activity. So what is the problem with this graph? Is there any problem?
Sir, maybe in Nagina 22 these genes express earlier,
Hmm.
or in IR64 this gene express maybe later. So the level of protein may be same, but expression is different.
No, what I am saying is one assumption is that your expression is directly proportional to concentration. So there is some problem with this graph. Can anyone say what is the problem with this graph?
Maybe there is some problem with the accumulation. Means the protein it is getting synthesized after
You
transcription.
remember this experiment is from the that experiment. Maybe you can assume that from this experiment the sample was taken. So what is the problem with this graph? Is there any problem? That is the reason. So when you express on the fresh weight basis, you see when you impose water stress or salt stress, the leaf water content varies. That is, the relative leaf water content will be different in different genotype. Between control and stress, there will be difference. So if there is a difference in the relative water content, which means when you take 100 mg of this one tissue, in this you may have about 70 mg of water, and here you may have about 50 mg of water. So only rest only is the actual cells you are taking. So there is a difference in the amount of tissue taken per se, but if you see often in many cases we report any metabolite we measure and report per gram fresh weight. So but if you express the RWC is different is there, and when you express based on the dry weight of the tissue, that is actual amount of tissue you take, you see there is a clear difference in the tissue. So it is important when you measure metabolites, you use express on dry weight basis, especially when you are doing experiment with salinity stress or salt stress. So this is to why this phenotyping so much important. Critically imposing stress is important. Growing plant at similar condition is critically important. This is illustrated by this example. So this is two transcriptome study. One is in Arabidopsis they examined the cold induced transcriptome. The cold stress may be one of the simplest stress to impose. In this particular paper, when they imposed cold stress, about 900 genes were differentially expressed. In another study also they did an experiment where the Arabidopsis plants were exposed to cold stress, and they also found about 800 genes they are differentially expressed. So if you see these two experiments independently, you will see that the number of genes that are differentially expressed under cold stress is about 800 to 900. So which is okay. It looks highly consistent. But when you compare the actual genes, like what are the genes upregulated or downregulated here, and what are the genes upregulated and downregulated here, only you find about 242 genes that are common between these two studies, although the experiment they described it is same. So which means that the way the plants were grown, or the rate at which the temperature was decreased in this experiment, these minor details normally we don't give in the experimental protocol. So only about 30% of the genes which we identified as cold responsive are actually cold responsive. Rest of the 60% or say 65% of the genes which we identified, they are nothing to do with the cold stress. So that's why it is very critical when we do experiment to conduct a proper experiment. In the temperature stress experiment also often we ignore an important factor. So this is an experiment which was say a hypothetical experiment I have given. So the heat shock protein expression is say strongly associated with the tolerance of the genotype. So this is a genotype of wheat which is highly tolerant to heat stress. This is another genotype which is sensitive to high temperature stress. So when the experiment was conducted and the HSP expression was studied, so under the stress condition they found that the expression of HSP was strongly induced in HD2329, but not in case of 3765. But although we know that this HSP is very important for stress tolerance, it is only an example of HSP. It may be any genes. When we subject the plant to high temperature and we study the gene expression. So actually this genotype is stress tolerant, it should have expressed more HSP when compared to this genotype. So but it did not happen. So what could be the possible reason, or is there any problem with this experiment? They have grown the genotype and then just put this plant in the greenhouse condition and exactly given the temperature. So is there any problem with this kind of experiment? You grow the plant and keep the plant in either in the temperature controlled greenhouse or in the growth chamber, and then you collect the tissue and do the differential expression study or transcriptome study. So whether it will give a real picture of the genes that are actually involved in the stress tolerance or not. So any points on this? I said this experiment, or they measured the temperature of the growth chamber. It was perfectly, it was all right in control and stress, but it was, the results was confusing. It is only for one, not only one gene, for several genes you find the same results. So what could be the possible thing they missed to measure? So one of the simplest thing they missed to measure is the temperature of the tissue. So when you subject these plants to high temperature stress, we assume that the plant experienced high temperature stress. So this is the genotypic variation. If you see, when you subject this plant to, say for example, some X temperature, some plants will have a temperature which is about near to the ambient temperature, and some plants will have a temperature which is very low when compared to the ambient temperature. So this genotype has a 6 degree temperature, tissue temperature, which is lower than that of the ambient temperature. This genotype is having the temperature almost similar to that of the ambient temperature. Similarly, you can find variation in any crop you take. The plants have an ability to cool their canopies by transpiration process. So if the transpiration process per se are different between the genotype, or the available soil moisture in the pot, if it is different between this genotype, they will affect transpiration and they can alter the canopy temperature. So whenever we do experiments, especially for gene expression studies, we should really see what is the temperature of the canopy rather than seeing the temperature of the chamber. So we will identify, like, just if you use this as a criteria, maybe if we have not known that this is tolerant, this is sensitive, maybe we will identify this kind of difference is there, so we may think that this genotype is better. Or we identify some genes which are not relevant to high temperature stress that are expressed in this, that are not expressed in this, and then we consider it as important gene for stress tolerance. So when you do canopy temperature, it is always important to measure the tissue temperature, and if it is similar tissue temperature, if it is there, then you compare the transcriptome of those plants, then you will see really the genes that are actually working under that temperature condition. So you can see a lot of variation between the tissue. It is like panicle versus leaf temperature is given. So there will be a lot of variation within the plant. So you have to critically measure this tissue and compare their temperature. Similarly, for any kind of stress, yes, please.
Sir, in case like here too, we can thermal imaging, we can stress measurement the canopy temperature. But in case like if is not the facility stability of this thing, thermal temperature, how can we measure the leaf canopy temperature?
So this one, if the thermal imaging is not available, you can use infrared thermometer, which are very cheap. You can get one infrared thermal gun costing less than about 15,000. So some measure, because we will be doing a transcrotom experiment which is costing several lakhs. So when we compare to that, these equipments are cheaper, and also we have to take utmost precaution to provide same amount of soil moisture, and the plants are pretty uniform, and then you expose the stress and also measure the temperature of the plant tissue before taking the sample. So the infrared thermometer is a cheaper one, you can use that one. Only when you use infrared thermometer, there is a problem of the field of view. So if infrared thermometer can measure, like if you focus like this, whatever object is there on the front, it will measure the temperature. So each infrared thermometer will have a field of view. So it may be smaller or larger depending upon the instrument, and also from the distance from which you are measuring. So try to see that the field of view is covering the plant tissue, not the pot or soil and other parts of the area. Yes.
In previous years, they did transcriptome analysis. Whether they did single transcriptome analysis or throughout stress period they did transcriptome analysis. Because in single transcriptome analysis, we cannot conclude that this gene is expressing during stress time. It should be throughout stress period.
No, see, all the genes, they have a different expression kinetics. Like some genes will be expressing early, some genes will be expressing late, and some gene expression will be sustained, and some may be like decreasing.
Gene expression is highly depend on environment and tissue specific.
Yeah.
That's why I'm asking.
Yeah.
It expressing throughout the stress period or initial single transcriptome analysis in each state we cannot conclude.
So that is true. We have to, for any gene expression analysis, to know whether it is important or not, at single time point or single tissue will not be sufficient. So you have to do at multiple, any gene expression, it is having kinetics, like it will start expressing and it will reach high, and then still even though if the gene may be required to function at further hours, since it has sufficient protein, till the half-life of the protein is, as long as the half-life of protein is longer, maybe that gene is shut down for some time, and once the protein is less, again it will be induced. So we have to do all this analysis at multiple time points. That's why it is very difficult to identify genes based on the transcriptome analysis or just gene expression analysis. Most of the genes identified through other approaches are better, like based on the genotypic differences or the mutants. If you identify genes that are more easy to associate with the phenotype, otherwise you will identify several and you have to validate their function. So I think next we will go to the mechanism. I thought instead of putting mechanism for each stress, I will give a general idea. Then most of you might have studied stress a lot in your studies. So when stress is there, so the mechanism of stress tolerance or the yield protection under stress can be put it as stress escape. So this was already exploited in plant breeding or by crop management. Stress is there here. Stress, for example, I have shown that here instead of plant I have put a like iron beam, and it is placed on two pillars. So assume that if the stress or the weight is falling on this iron beam, the iron beam is going to bend or break. So similarly, if the plant experiences stress, it is going to give less yield or it is going to die. So if the stress or if the weight is not falling on the iron beam, then there is no harm to the this iron beam. Similarly, if the stress is not occurring in the phase where the plant is grown, then there is no problem for the plant. So if you want to grow a crop in the Kharif season, if it can mature in Delhi, say if it is in 60 days, if it can mature. If you do sowing in July, by August end you will harvest. So you know in Delhi you have rain for 2 months, so the plant may not experience stress. Or you do it in the stored soil moisture condition, like in the winter you do a short duration crop, maybe the soil moisture is sufficient to meet the requirement of the crop. So most earlier stage of plant breeding were focused on this one. They try to match the season and the rainfall pattern with the crop, and they try to develop short duration varieties. Because of that, the yield was stabilized. But this is often not sufficient mechanism, or there is no mechanism, a biological mechanism is involved here. Actual biological mechanisms, when the the plant faces the stress, what happens, that is the actual biological mechanisms which we are interested in. Say for example, if the weight is falling on the beam, so it can have different responses. So instead of 2 beam, so if there is a 3 beams are there and the weight is falling on this particular place, so it may not have that much impact. If the weight is falling on this beam, so because there is no support from the bottom, so it may have some bending or breakage. But if it is weight is falling on this or here, or you have wherever some support is there, it will not have any effect. So these kind of mechanisms are also exist in plant, where the say for example a plant is having a deep root system, so when there is no rain it can take up more moisture from the deeper layer of the soil, so it will not experience the stress. Or it has higher transpiration, so when you subject them to high temperature stress, they may not actually experience the tissue temperature. Or in case of salinity stress, it has ability to put all the salts into vacuole, or it can secrete in the secretory glands, so they may not face this stress. These are some of the avoidance mechanism, constitutive avoidance mechanism. Some of these may also be induced, like in some cases the plants may have deeper root constitutively, that is by birth they have that character. Sometimes when there is a mild stress starts, the plant senses that stress and then they can grow the root deeper. So these kind of cellular mechanisms are very important. These are called as cellular stress postponement or avoidance mechanism. That is, there is a environmental stress is there, but the plant is postponing the stress in the tissue by using mechanism that helps in postponement or avoidance. So when this mechanism, first all the plants they try to use this mechanism, and when this mechanism is facing the limitation, then the cells experience stress. So the plants have several stress tolerance mechanisms. So once even though there is some impact, that is visually you can see the iron beam is bent, so similarly the plant may be it might have reduced stature, or some of the leaves are dried because of the stress. But if it has cellular stress tolerance, it can have better recovery. That is, some of the responses are elastic, that is under stress condition the process is almost broken down, but when the stress is relieved, these processes are recovered to its original states. Some of the mechanisms, they are plastic, although they will recover to a certain extent, but still it will be less than that of the normal plant. So these recovery mechanisms and the cellular stress tolerance mechanisms are very important for improving the stress response. So for each of these stress, if you think you can identify various mechanisms that have been identified, you have to put into these categories and see in the target population of environment what kind of mechanism operate, and based on that you can select genes for genome editing. So, so far we talked a lot about environment, and we also should know what is the mechanism of stress tolerance and what are the genes involved. So again, this is a very vast topic for each stress to discuss what are the genes involved. So any stress is that it is going to produce some signal. How the plant will know? It has to have some physical, chemical, or electrical, or some redox signal. Some signal should be there from the stress. So if water is not there, a plant get what signal? Signal is what? It's wilting is a response. What is the signal? closing of stomata is an response. ABA is okay, it is a secondary signal. So immediate, it one is like when the water is not there, water evaporates from the cell. Cell is like plasma membrane is there, cell wall is there. When it is having water, fully water, the plasma membrane is oppressed with the cell wall, it is turgid. So when water is not there, water is evaporating from the cell and it is not getting water from the root, so it shrinks. So that is the turgidity loss or the osmotic potential change is the immediate physical stress the plant feels. So that can induce some signal, that is membrane is detaching from the cell wall. So some of the wall associated or membrane associated protein can initiate cellular process, or because of less water available there is a change in metabolism. Say if water is not there, like as you said, the stomata closes, but still light is coming, the cell is having the same amount of light. So it will produce lot of electrons that will lead to production of superoxide and other radical, or say the Calvin cycle is stopped, so the metabolism is affected, so many metabolites can be produced. So large number of signals can be there, it may be primary signal or other kind of signals can be there. So there are sensors, and we have maybe very less sensors that directly sense the primary signals per se. So still lot work needs to be done how the plant senses various kind of stresses. So once they sense, often it can produce some sensor, it may be the membrane bound or it may be intracellular, it produces some second messengers, and the second messengers do the signal transduction, which you might have studied a large. And also it can produce secondary signals. Secondary signals are like ABA is not a like direct signal of the stress, it is a secondary signal. Once the stress is there, the plant produces this secondary signal to amplify the signal transduction. That is either the inactive ABA is activated by beta glucosidase, or the ABA synthesis is promoted and actual ABA amount of ABA accumulate, and these kind of secondary signals can be produced, which will amplify the signal, and these signals can be mobile from one tissue to other. Like for example, if root faces stress, it produces ABA and it sends it to the leaf to close the stomata. So these are kind of, yes? Electrical signal, it is produced in all kind of stresses. So only like when what is electrical signal? So the membrane potential is often minus 150 millivolts something, it is membrane potential. So any stress that affect the master enzyme, what we call as the proton ATPase, which is present on the plasma membrane, if any stress that affects the ATPase activity, it is going to alter the membrane potential. Or if some ionic stress is there, some outside, it is like some ions are there which is taken inside, and if it is reducing the membrane potential, the potential difference between the membrane, that can cause electrical signal. So this one has been studied, like often people have studied, but so far it has also been demonstrated that plant, when you give stress to the plant, it produces an electrical signal. So if you take, let's say for example, if you take a wheat plant, this is one of the studies conducted long back in our division under the guidance of Professor late Professor S. K. Sinha. So what they have done is they have taken the wheat plant as a fibrous root system. So you take the wheat plant, and half of the root you put it in normal water, half of the leaf you put it in the stress condition, that is you can use PEG or something. So when you measure they measured the electric potential when compared to the entire root system is in the normal condition, and half of the root system is in the PEG, or the entire root system is in the PEG. So when the half of the root system is in the PEG, the plant still it should those amount of roots are sufficient for taking the water. Like if you remove half of the roots, if you cut rest of the roots, then also the plant will not experience the stress. But if when these roots are, some roots are subjected to stress, they found that there is a difference in the electrical potential of the leaf changes, and then this lead to stomatal closure. So this has been studied, like several people have reported this, but still we do not understand like how it is propagated, and these mechanisms are being studied. So once the signal transduction is there, you get alteration in the protein stability or activity, and then that can directly like causes any physiological process. The protein is inhibited or activated, it can change its activity, or it alters the gene expression, and ultimately you see lot of changes, you get the quality and yield changes. So we have maybe several thousand genes so far known for all these things in maybe several crops, but our difficulty is to pinpoint which gene we have to select for editing, and how much impact it is going to have. So if we can select either the positive regulators or negative regulators for editing the genome. So if you are using different approaches, like if you are using positive regulator, then we need to enhance the expression, or if you are using the negative regulator, we have to down regulate the expression. So now we will see some examples of genome editing. This you have listened for the last several days, so I need not put much emphasis on what is genome editing. I will directly give some example. So this is one example wherein a positive regulator is upregulated by using a genome editing technology. So the Argos 8 is a negative regulator of ethylene signaling. So ethylene is one of the classical plant hormone which is induced under several stress condition and development, and if ethylene is induced, it will affect the reduce the root growth, and it will induce the cell death process. So if we can suppress the ethylene signaling, several evidences they have shown that we can promote the stress tolerance of the plant. So in this case, the Argos 8 gene was selected for this purpose. This is a negative regulator of ethylene signaling. And earlier study they showed that when you do the Argos 8 overexpression in the maize and Arabidopsis, and the maize plants were tested in the field condition, and they found that this gene can enhance the yield of the maize without causing the yield penalty under normal conditions. So when you select a gene for improving the stress tolerance, you should see that this gene overexpression or knockout, it should not compromise the yield under normal condition, but it should cause the yield stability or yield for better performance under the stress condition. So often in the farmers field, you may or may not get stress in some cases, like if it is drought, if it is rainfall dependent area, you may get good rainfall here and bad rainfall here. But for salinity, you know that there is going to be a stress. So depending upon this stress, you see that the performance is better under or equal under normal condition, and only under the stress condition the gene gives the phenotype. So in this case, they have selected this Argos 8. The Argos 8 gene expresses in the, this is initially they studied in Arabidopsis, the expression level is less, and when, so that's why they want to enhance the expression of this Argos 8 gene constitutively, so that the high level of Argos 8 can regulate ethylene signaling, and it can inhibit the ethylene pathway so that it can produce better performance under stress conditions. So for that they needed a promoter which is not very strong, otherwise the entire ethylene signaling may be inhibited. So they selected a promoter. This study was conducted in maize. They selected a GOS2 gene promoter which has a moderate expression. So what was done was, in this they used homology dependent repair pathway for editing this Argos 8 gene, that is to enhance the expression of Argos 8. So the native promoter is weak, so expression is not very high. So they used the GOS2 promoter from the maize only. So the same plant they took a promoter region from the GOS2 gene and use two different guide RNAs, one guide RNA, or they used two different guide RNA to replace the Argos 8 promoter, or they used one guide RNA to insert this Gauss2 promoter in the 5' UTR of the argoside gene. So in this they use homology of about between the after the promoter, the flanking region where about 400 base pair homology was used, which is homologous to this region and this region. And by using the homology dependent repair pathway, they could develop both the kind of plant, that is Gauss2 promoter is inserted in the 5' UTR in the beginning of the 5' UTR, and then it can drive the gene expression, or they replace the argoside promoter with Gauss2 promoter. So both are from the maize. So it is a kind of promoter replacement within the plant, a cisgenic approach you can say. And they found the expression of these plant when the promoter was replaced, they found that the expression was higher when compared to the wild type plant, or it is only inserted in the UTR region. And they compared the yield performance of these maize plant under the field condition. So when the flowering stage stress was there, they found there is a 6 bushel per acre difference was there, which was statistically significant. Although there was under the grain filling stage stress, there is no statistically significant difference, or under the optimum condition also you find the difference is not statistically significant. So you can do this kind of approach, that is you modify the promoter to regulate the expression of a positive regulator, and then it is possible to enhance the yield.
Sir, I have like one.
Yeah.
Sir, this thing, promoter is always fascinating to me to understand this thing.
Okay.
So in case of promoter, how can we like regulate the expression? How much knockdown we want to do? There is some specific elements there, or we just select by the weak promoter, strong promoter in that case?
So in this case, they selected a promoter which is having a moderate expression, and then they put it. So it depends. Like promoter editing is really very good. It can be used in a wise way in especially in the plants, because when you knock out the gene, we don't know why the nature has selected that gene. It might have some function. So unless otherwise we are not very clear, the knockout may have some impact. So you need lot of studies to see the gene is really not important. So otherwise the promoter editing is very good, because this gene, if you study many of the stress responsive gene, they express in some tissue it is highly induced, in some tissue it is not induced. So you can identify the promoter elements that specifically drives the stress responsive expression. You can leave the constitutive expression. Like if you delete say ABRE binding element in a drought responsive gene, only the drought responsive expression will be reduced. Otherwise the rest of the expression still the cis elements are intact. So the regulation by other pathway, developmental pathway, they will be still, it will be working. So that's why we need lot of understanding about the promoter. It was shown for the bacterial blight resistance in rice, the sweet genes, they are the sucrose transporters. So those are also essential for the plant function. What they have done is they have edited the tail binding sites in the rice. So that paper I have not put it, but somebody will discuss when they discuss the disease resistance, or you might have read that paper. So wherein the tails, they bind to the sweet genes and they induce the expression when the infection is there, lot of sugar is exported to the apoplast, so the bacteria can grow better. So what they have done is they identified the tail binding sites, it is known, so mutated only that cis element. So the rest of the expression is maintained normal, only when the bacteria infects, the expression is not getting upregulated. So if you have understanding of the gene, then only we can go for what to do it. So this one is the another kind of examples available. There are several examples available in stress tolerance. Many of these examples are like when you knock out the positive regulator, they give susceptibility. So I did not take many of those examples, only one I have taken for that purpose. And most of the other examples are when you do CRISPR genome editing, you find the phenotype, you get alleles which are so totally novel function or totally different function. So the cytokinin, it is very important hormone. It is anti-senescence hormone. It is important for stress. Cells should maintain certain amount of cytokinin. If it too much cytokinin, then plant is sensitive. If it is too less, the plant is not growing. It is going to die. So some amount of cytokinin is important. One of the group of enzymes that are important for cytokinin biosynthesis is the lonely guy enzyme, which is downstream of the isopentenyl transferase. Most of the studies have been conducted with overexpression of this gene. So what these people have done is they screened almost 2 lakh activation tagged lines of rice. They identified a drought tolerant plant. And when they did the flanking sequence analysis, they found that tDNA insertion was there in the 3' end of the this lonely guy like 5 gene in rice. So the mutation was at most at the end of the coding sequence, and but this plant was tolerant to drought stress. So they want to use genome editing to create some allele and study their stress response. So they targeted a region which is marked here around 225 to the latter portion of the region was targeted by using 3 different guide RNAs, and these are the kind of mutants they obtained. These are the nucleotide sequence comparison, though they grouped depending upon the nucleotide deleted, different kind of edit they grouped, and this is the kind of protein change. If there is single base pair or 2 base pair deletion, and there is a frame shift, and different kind of proteins forms after this. Up to this, the protein is same as that of lonely guy 5. Then after that, there is a difference in the sequence between the wild type and different edited lines. So you can by doing this mutation, you create large amount of allelic variation. So you can consider each of them as a different kind of allele. And the interesting result was when they analyzed the stress response. You can see they analyzed for the 2 stresses. One is the nitrogen deficiency stress, and another one is drought stress. When this particular gene was overexpressed, that is, this is constitutive overexpression by using transgenic approach, actually the yield decreased in both normal nitrogen condition as well as the stress condition. And these values are significant differences. If it is less than 0.05, you think that the difference is significant from the control. Some of the edits here, you can say that these edits, edit B, edit C, they had better grain yield under the normal nitrogen condition, as well as under the stress condition also they produced better yield. Similarly, under drought stress condition, these edits, these are experiment conducted in the field in 2 different places in China. And here also they found that some edits they performed better under stress condition. This in one experiment they have got about 26% to 27% yield advantage in some of the edits, whereas other edits they are not providing that much tolerance. So this not only it helps in producing this approach, CRISPR approach helps in producing the stress tolerant plant, but it also helps to engineer a lot of alleles which are maybe naturally present, which we may not know, and then to understand the function of that particular gene. The other examples, some examples I will give from the abscisic acid signaling pathway. So abscisic acid is one of the plant hormone which is studied almost from 1960s. Large number of people have worked on this, and it having several roles, from the stomatal closure to normal vegetative growth and seed development and seed dormancy, and the root promotion, extreme stress tolerance. This under abiotic stress, it is almost having a promoting role, and for normal vegetative growth it needs certain levels of ABA. If ABA is high, then also growth is less. This is a ABA1 mutant where ABA is less. If ABA is less, then also growth is less. And for seed development and dormancy, ABA is important. If ABA is not there, the seed is going to germinate on the mother plant, so we will have great difficulty in agriculture. But in case of biotic stress response, there are like the responses are different with different kind of pathogen. So this is with for this rice brown spot disease, the ABA is a positive regulator. For some of the other diseases, it has been shown as a negative regulator. So it has a dual role. So when you work with ABA pathway, you have to see how to balance between the biotic and the abiotic stresses. And ABA is also important nutraceutical, because this is not only a plant hormone, it is also an important hormone in human beings, where it controls from innate immunity to the insulin response in human beings. So that is, it is a pro-survival factor in plants also. So the ABA signaling pathway, at least the core pathway, is well established now. So you know what are the positive and the negative regulators. So the receptor, at least one set of receptor, they are soluble receptor. They are present in the intracellular space, that is in the cytosol. And when ABA is not there, the receptor is alone. It is not interacting with any of the other protein. So when the ABA is not there, the protein phosphatase 2C and clade A group of protein, there are about 9 to 10 genes are there in different plants. And these proteins, they interact with the SNRK, that is SNF1 related protein kinases. These are serine protein kinases. And two different families, SNRK1 and SNRK2 family proteins, they interact with this protein phosphatase 2C. So this protein phosphatase 2C bind to this kinases, and it is not allowing the function of the kinase. It continuously dephosphorylates these kinases. So these kinases are autophosphorylated, and then these kinases transfer the phosphate group to, or it phosphorylate it targets. So that is important for inducing the activity. So these SNRK1 family kinase, they are involved in the energy signaling, whereas the SNRK2 kinases, they are involved in the stress response. So when there is a stress, ABA accumulation increases, and this ABA now it binds to the receptor. Once the receptor, the ABA binds to the receptor, the configuration of the receptor changes. It produces a better configuration which can interact with protein phosphatase 2C. So now the protein phosphatase 2C is sequestered by its interaction with the ABA receptor. So now the kinase is free. So these kinases then can phosphorylate variety of transcription factor, or these are some of the ion channels or the aquaporins. So many membrane proteins can be phosphorylated, and some of the phosphorylation can inhibit the activity, some of the phosphorylation can activate the activity of protein, and thus you get the stress response. So in this, some of the negative regulators are the protein phosphatase 2C is the major negative regulator, and in addition to that, it was reported about 20 years back, the farnesyl transperases, they negatively regulate the signaling, but we don't know exactly where they work. But these are a group of protein which work as negative regulators. So these two are the important negative regulator of the stress signaling. So either you can modulate the receptors, or the positive regulators, that is the target the transcription factor, or you regulate this negative regulator for improving the stress tolerance. So when you want to target the positive regulator, either you can enhance the expression by, as I told earlier, they used the promoter replacement approach to enhance the expression. Alternatively, the other approaches can be URF editing, that is upstream URF regions are there in often about almost 30 to 50% of the genes coded by different organisms, they have upstream ORF, which is a short protein coding region. So that often hinders the translation process, so the protein produced from that transcript is less. So either you can modify that, or you can modify the promoter, or you can modify the epigenome, or in that particular region, so that expression is enhanced. So in this approach, they used they modified the epigenome of the promoter of the ABF2, which is one of the important transcription factor that regulate several stress responsive gene. So in this, the D-Gas9 was fused with histone acetyl transferase, which can acetylate the histone 3 lysine 27 residue. That is, once it is acetylated, the chromatin is decondensed, so it is easy for the enzymes to access the region for transcription process. So they used two different guide RNA to guide the Cas9 and histone acetyl transferase to the specific location. And then they developed, initially they developed with these two guide RNAs, they developed transgenics, and they found that in these lines the expression was better. And then they also studied their stress response and stress responsive gene expression. This is one of the RD29A is one of the stress responsive gene which is regulated by this transcription factor. So they found that under stress condition in the transgenic the expression was high, and they just done a small study to see if it can enhance the stress tolerance. So with the plant with guide RNA, they found that the plants are more tolerant to dehydration stress, whereas the plant without guide RNA, they are not tolerant. So this is after dehydration, the plants were rewatered and the recovery was analyzed. So this is an example just to show that this is an approach where you can modify the histone acetylation to enhance the stress tolerance. But however, this will be considered as transgenic approach only because this transgene is always, it should be there in the plant for keep activating the expression. Besides this, this ABA receptor family in Arabidopsis in other plants, it contains large number of genes, unlike other hormone receptor where few receptors there in each plant. ABA receptors, one particular group, that is the soluble ABA receptor, large number is there, are more than 10 receptors you find in many plants. So in order to study their function in stress, and in order to see that, because these ABA receptors from the Arabidopsis studies, it was known that some of the receptors were important for the regulation of stomatal control, and some are important for germination, some are important for both the process. So in order to study these ABA receptors in Zhengkang Zoo lab, they did mutagenized all the ABA receptors by using the CRISPR-Cas9 technology, and also they obtained single, double, triple, and all kind of mutants in different mutant combination, and they studied their yield performance. So this is one of the studies where if you mutagenize these ABA receptors, you find that these plants, they grow better under normal environmental condition. That is, the growth when compared to wild type, this plant growth is better and they produce higher yield. But we have to see, identify the plants or identify the mutant combination. It promotes the growth under normal condition, but it should not cause other problem. As I told, ABA is important for vivipary. So this is the mutant where you can see all the seeds are already germinated before harvesting. Still the leaves are green. You see all the seeds germinated here, whereas not in the wild type. So in these kind of mutant combination they analyzed, and they found that some of the mutant combination, that is file 1, file 4 and file 6. When this mutant combination is there, the viviparous germination was very less. So this is, if all the genes are mutated, you get lot of viviparous germination. If only these 3 genes are mutated, they found the viviparous germination was less. And then they also analyzed the water loss to see how it is going to affect stress response. So in this case, here if you have mutation in many genes, the plant is going to lose more water. But if you have mutation in few genes, maybe the plant is going to lose water in an intermittent way. They examined the response of these mutants, mainly the triple mutant. They studied which were appear to be promising, that is less compromised in the viviparous germination. They performed better, and they produced about 25% higher yield under normal environmental condition in 2 different years of study. And they also analyzed the transpiration, and as expected, because these plants are unable to control the stomata, you find more amount of coolness and they produce better transpiration. However, if you are subjecting them to any kind of water stress, these plants are going to die. So this study, it shows that like these plants, although the mutagenizing ABA receptor can be one approach to enhance the yield under normal condition, but if there is any stress, then it is going to be disastrous. The other one, it is so far people have not tried. I am putting this approach for you people. So this one is the Arabidopsis, the ABA receptor. As I told, it is ABA is very important for stress tolerance, but the excess at non-stress condition, it is going to be detrimental. So once the ABA receptors were identified, people were using different approaches to improve the stress tolerance. So one of the approach what was tried from maybe 1970s is that whether you can immunize the plant, that is you spray ABA to the plant before stress, the plant is ready for experiencing the stress. So that works very well with the plant, but ABA is costly and other problem is there. So that's why it is not sprayed in the field condition. So if you can identify naturally or the chemical that is used in the agriculture normally, and if you can spray that chemical, if you can modify the ABA receptor to bind to that particular chemical, then you can easily identify a chemical which is readily available in the market, and you can just spray that, which is also cheap, and the ABA response can be mediated. So this kind is possible with the engineering of the receptor, which is called as orthogonal receptors. So these receptors, you modify the native receptors, you edit some of the amino acids which bind to the native ligand. You modify the binding packet to of the receptor so that this receptor can bind to a non-native chemical, but it will not bind to the native chemical. So what they have done is, in the ABA receptor, one of the ABA receptor, they made 6 different amino acid edit. So now this receptor can bind to one of the fungicide which is used to control the phytophthora. The mandipropamide can now bind to this receptor, but this receptor cannot bind to abscisic acid. So whenever they mutagenized this receptor, and then they produced a transgenic plant. So you can also do this by homology dependent repair in non-transgenic way. So this is a transgenic plant wherein these mutations were created, and when you spray this fungicide on the plant, this plant closes its stomata. You can see this one is the thermal image, and if the stomata is closed, you can see a rise in temperatures. When they sprayed on this Arabidopsis or tomato plant, it closes stomata, and also it gives excellent stress tolerance. The wild type plant sprayed with mandipropamide before exposing to the stress almost died, whereas these plants, transgenic plant, sprayed with this mandipropamide, they are tolerant. So this is possible, like we can use a CRISPR approach to make these kind of edits in the receptor, so that you can engineer orthogonal receptor in the plant, and you can use the agrochemical for control. So earlier to this, there are several agrochemicals or several chemicals which are not commonly used were also identified for this purpose, but the regulation is another issue. Like transgenic, even any chemical, if you want to register for agriculture use, you have to have the environmental safety, human toxicity, several tests need to be done, which is even costlier than the regulation of GM crop. So that's why this kind of approach people are trying. So whatever the already approved chemical is there, so if we can modify the receptor so that through modified protein, you can use the native chemical for regulating the stress response. The another approach from the literature it was there was on the reducing stomatal density. So if you reduce the stomatal density, you expect less amount of transpiration. So by using both CPF and Cas9, a study was done in IARI, and they generated mutant, and they found mutant with drastically reduction in the stomatal number. But in this paper, the results on the stress response or the water use was not there, but it is an approach that can be used to reduce the stomatal number. So you have to see what is the critical number you should maintain. You cannot like totally shut off the stomata and then you produce any yield. So this is another approach people are trying to target. So in our lab, we are from the last year, we are also working on some of these genes, drought and salt tolerance, and the microRNA 169, protein phosphatase 2C, farnesyl transferase, CBP20. These are the stress-related genes we are working with. In one gene, we have made some progress. In other gene, the work is in progress. Some mutants have been identified in some of them, and in some of them, it is in the process of development. So one of the gene which we selected was the drought and salt tolerance. Not only we are working, even many other people may be working with this gene, because this gene was a popular gene which was identified in 2009, which gives tolerance to both drought and salt stress, extreme drought and salt stress. This gene was identified again based on the mutant screening. So they screened about 270,000 EMS mutant to identify this particular mutant. So we also screened large number of the Nagina22 mutant, but we could not get this allele from that one. So that's why we have selected a genome editing approach. So this is a this gene, the drought and salt tolerance gene was identified in 2009, and this protein regulates the stomatal control mainly through the hydrogen peroxide homeostasis. So in stomata, you know, the respiratory burst oxidases are activated by ABA and other signaling, and they produce hydrogen peroxide, and then hydrogen peroxide plays an important role in stomatal closure. So this gene in an ABA independent pathway, it regulates peroxidase 4. That is, if the normal version of this transcription factor is there, it binds to peroxidase 24 promoter and it induces the gene expression. And if the peroxidase expression is high, it is going to use the hydrogen peroxide, and the hydrogen peroxide signal will not be there. So it is a kind of negative regulator of hydrogen peroxide accumulation. So when this gene is mutated, in this particular case, the DST1 mutation was there in the N69 was converted into D. Asparagine to aspartic acid mutation was there. That was two mutations they identified, but this mutation, by using the molecular studies, they confirmed that this is the causal mutation for the DST mutation. So if there is a mutation in the zinc finger domain, this protein is unable to bind to the DNA, and then so it is not able to regulate, induce the expression. So these plants are more tolerant to drought and salt stress. Besides that, there are several other studies. They showed that this protein is an important candidate. That's why we have selected this one. They also from the microarray study, they found that one of the kinase was highly induced in the mutant version. So based on this, this one is like induced by DST gene when it is overexpressed. So they identified this particular mutant, and they also validated that is the kinase which is interacting or which is downstream for the DST gene. When the this gene is overexpressed, then the plants are sensitive. That is, if there is mutation in this gene, the plants are tolerant. If there is overexpression of its target protein, the plant is sensitive. So it suggests that this is a good candidate. And by using Yeast2 hybrid also, they identified another one co-activator zinc finger protein, which is called BCA. When this plant was overexpressed, then also like these plants produced a sensitive phenotype to drought and salt. So by different means, it appears that if we mutate DST, we may get good phenotype for stress tolerance. In addition to that, the earlier study was also shown that they identified from the different aspect that they want to know the plants which can produce higher grain number. Again, EMS mutant screening led to identification of gene which they named it as regulator of GN1A. GN1A is gene for cytokinin oxidase that control grain number. So in this case, there was a mutation here. A single base insertion was there between these nucleotide, which results in the frame shift mutation. So if we see the normal DST sequence versus the reg1 sequence, the amino acid sequence is same up to this one. After that, there is a frame shift, and then the rest of the sequence got changed because of the single insertion mutation. This led to increase in the grain number. So we thought using this will be a very good approach for producing both stress tolerant version as well as high grain number version. So we developed two guide RNAs for targeting two different regions of the DST gene, and we got large number of alleles. Some of them deleted, like deletion mutation, where the entire region between this to this place is deleted, and we got this deletion mutant. In addition to that, we have also got insertion mutant, whereas there is insertion here. After that, you see that the entire protein sequence got changed. So you have got all kind of alleles that can be analyzed for the stress response as well as yield increase. So as reported from the EMS mutant, when there is a mutation, loss of function mutant, that is N16D, which made the protein unable to bind to the cis element of the target gene, that gave drought and salt tolerance phenotype. And that mutation also, they have reported that the enhancement in the leaf width and reduction in the number of stomata for plant. So in the deletion mutant which we generated also, there was a significant increase in the leaf width and a significant reduction in the stomatal number, and there is a reduction in the relative water content when it is subjected to excess leaf water loss. And the salt stress in the seedling stage we have tested so far. So it gives a very high salt stress. This is 200 millimolar salt is very high for rice. The wild type plants, they died, whereas these seedlings can tolerate the salt stress. So these are some of the results. In other cases, we have in different stages of development. As I told, we have also targeting the protein phosphatase 2C and the farnesyl transferase, because earlier studies they have shown that although not in rice, but in Arabidopsis it was shown that if you make mutation of the protein phosphatases, you can produce a stress tolerant plant. And the farnesyl transferase, by using antisense approach, it was demonstrated long back. So in the field condition also it can produce higher yield. And also this another gene, nuclear factor Y, when it is overexpressed, it can give still stress tolerance in the field. And one of the study reported that the microRNA 169 is controlling the expression of the nuclear factor YA. So that's why we selected the microRNA 169 for editing. And also we are working with cap binding proteins. These are involved in the mRNA processing, and they are also involved in the microRNA biogenesis. So there are two important cap binding proteins are there. One is the cap binding protein 80 and another one is cap binding protein 20. So when there is a mutation in these proteins, and the plants, they perform better under drought stress conditions. So we are also working with these genes in rice. So these mutants are in different stages. So these are the different approaches. Either you take an allied genotype and you mutate some of the genes for stress tolerance, and then see if you can produce a stress tolerant plant. An alternative approach is that there are several plants, they are naturally stress resistant. Whether you can mutate some of the genes to induce the domestication trait, so that you have stress tolerance is there, you get the plant which is accepted by the consumer. So this is the pokali rice. You see it is cultivated in the seawater inundated field. It is highly salt tolerance, but the grain quality, many of the people may not like the grain quality. So whether like this, you have several genotypes which are highly stress tolerance, whether you can modify some of the genes, either improve their quality or yield, whether you can use them for the purpose. So far, like there are not many any examples are there for this purpose except for one example. So there are several list of plants are there which are naturally stress resistant, whether you can edit them for improving the stress tolerance is a question. So this you might have you might have working with genome editing might have studied the de novo or accelerated domestication publication which came in 2018, wherein few genes have been edited in the wild tomato Solanum pimpinellifolium to enhance the yield of this plant. So this plant is tolerant to salt stress, and this plant is tolerant to this disease resistance. So they by editing these different genes, they produced a phenotype which is maybe somewhat near to the domesticated plant phenotype, but when they analyzed the stress tolerance, they maintained the level of disease resistance which the Solanum pimpinellum folium had, and also the salt tolerance which the plant had. So this is another approach wherein you use a plant which is naturally stress resistant and use edit genes which are not related to stress to produce a plant which is highly stress tolerant. So these are a summary of the different approaches that is used in the genome editing. Use either gain or loss of function mutants by using different editing technologies, or use the base editing. You can also regulate the transcription by either the promoter editing by creating inserting some elements, or you do base editing to modify some of the cis elements. By this approach also you can alter the gene transcription, and also you can regulate do the post translational regulation, that is the transcription is not affected but the translation product may have a different impact, where you can alter the splicing sites, that is by base editing can be used to modify the AG and CT so that the splicing is altered, or you can modify the upstream ORFs, the small ORF before present before the actual initiation codon, that will enhance the translation, or you can put some of the translational enhancer in the genes for improving the stress tolerance. So to summarize, stress tolerance can be improved, but you should understand it properly. And for example, drought tolerance you can improve. You have to make a consistent effort, that it is not that one shot you will get everything. But we should remember that when you work with the essential environmental factors, you cannot produce grains without the input. That we should always remember. That many people say drought research is going on for several years, nothing has happened. So it depends like what is the kind of stress. We cannot say nothing has happened. So some significant improvement is there even in the field condition. Genotype which use less water have been produced, but we should not expect too much, that is every output requires some input. Without input we should not expect output. So the regulation part, I think it will be covered by Dr. S.R. Bhatt in the coming days. So this example you might have studied. So far only the calyx high oleic acid soybean is the like edited product which is the cultivated in USA, but this is edited by using a talent technology to produce high oleic acid. But so this gives hope that other genome editing product can be used. In some countries there is no regulation, you can use the genome edited product free, but in our country there will be a regulation, and this will be discussed by Dr. Bhatt. So last but not the least, it is the question that gets you Nobel Prize. So after that, you just add lab coats, coffee, or air conditioning. So you ask the right question. That is very important. Other things are all the tools are available. You can, even if it is not in your place, you can access from the other place, or you can go in other place and work. But we should ask the right question. For that, we should read a lot and think a lot. And finally, I thank all my lab people who are working with me in the genome editing as well as in the other project, and also the funding support from our institute as well as the National Agriculture Science Fund of ICAR and DBT Government of India, which has funded our work, the current projects on genome editing. And another one important Nobel Prize sentence I always use in my slide is, one who solves the problem of water is worth 2 Nobel Prizes, one for science and one for peace. So when not only when you work with the stress, and otherwise also we try to conserve water, because water is a precious resource which is decreasing day by day. So we should work towards saving the water. So that's all from my side. Although I have taken longer time, I thought I will give that introductory part, which is more important rather than the latter part of genome editing. In any case, you are listening from all the speakers. So I thought we should understand the stresses and stress phenotyping properly, so that we can produce better results. Thank you for your passionate listening for almost 2 hours.
Thank you, sir, for the excellent talk. Any more questions?
Sir, thanks for delivering the wonderful lecture. I have a question regarding the stress tolerance. We are trying to enhance the tolerance to overcome the stress. But there are the some spaces in which stress is beneficial. Like there is a wild flax in which
Wild?
wild flax.
Okay, flax, okay.
Wild flax. It is a case in the botany, and wild flax is able to overcome the there is a type of the soil, serpentine soils, in which the calcium is in the lowest amount, and this is one of the aspect in which it is going to the toilet. But there is a benefit that it is help to adapt in the to provide the decline in the pathogen resistance. What we can do in this aspect in which the stress is beneficial and harmful also.
So, see, if the stress is beneficial, it is good. No, you can cultivate
Yeah.
that crop in those area. But in those crops, as I told in the last part of the slide, maybe those many plants, they are resistant to stress. And whether you can improve the economic yield in those crops. So we need not always work on the stress. No, we have to sometime work on the other part of the crop also. Like we can enhance the yield potential of those crop in those where they are already stress tolerant. Then we need not worry about it. Like now people are thinking that why only eat and rice, you grow cactus in desert and use cactus as a food, modify the cactus so that some nutritional values can be added. So when the human population is increasing, we have to explore all the aspects. We cannot say only improving stress tolerance is important. And also the stress tolerance, as you study more and deeper, you get like more and more questions. Often sometimes they may be tolerant to abiotic stress, but it may be sensitive to biotic stress. Same genes it may control, like the MAP kinase, if you take, who is working on MAP kinase, it is there MAP kinase, sometimes it increases tolerance, sometimes it decreases tolerance, even between the abiotic stress and between the biotic stress. So same gene can have diverse role, but in this case the genome editing is a boon where you can engineer lot of alleles. So you can engineer several alleles where you can make that protein work specifically under that condition as favorable, and other condition you can remove the non-favorable alleles or the regions of the protein that may interact with some of the other proteins. So that in that way you have a lot of opportunity for engineering.
Thank you, sir.
Sir, my question is regarding that receptor mutation in ABA receptor. So the nature might have evolved lot of receptors, so they may have some tissue specific or condition specific function in case of that receptors. So if we completely knock down that receptors, we should analyze all the other parts also, right? So because lot of other situations may also arise in case of plants.
So that's what these receptors earlier people thought that these receptors are redundant, they may have the same function. But these studies, they clearly showed that they every receptor, they have some or other essential role to play. In that paper, if you find, you study in detail like this paper and some of the earlier one paper where they overexpressed all the AB receptors. So these are studies wherein they completely analyzed all the receptor families. So you get very interesting phenotype when you overexpress, and when you do knockout, the single receptor may not have any impact at one particular stage, but in other stage it may have some impact. So in this, that's why you have to create all possible combination of receptor, and then we have to see what is good. Like although those receptor mutation, 3 receptor, this triple mutant, 146 triple mutant, it is and performing better under normal condition, but if any stress is there, it is not going to tolerate. Like if you are doing a controlled condition, say you are doing a vertical farming or precision farming where you are sure that there is no stress, then we have to use those kind of mutations also where it is possible, like where the VVPR is not an issue. So you have to see what condition, and then you have to select the gene. There is no single solution for the every problem. So we have to approach the problem bit by bit. Thank you.
Sir, I have a question in my PhD. So I am getting one problem. I need your suggestion in that. So I am working in some photoperiodic gene. So I am doing also stress phenotyping of that gene also. So what is happening? My transgenic getting early flowering in that case. So I want to expose stress, because as you mentioned that whenever a stress we providing, we have to make sure that each and every condition should be ideal, like same for the both like susceptible or transgenic in this case. So I'm getting early flowering. So at what time I should look down whether this time I should give stress, because in vegetative stage or reproductive stage, if vegetative stage we give before the panicle anthesis. So in that case, my transgenic is giving anthesis earlier than wild type. So how I can there I can impose this stress in that way.
So if it is what crop?
Rice.
Rice. So in rice, depending upon the crops, about it takes 30 days when you see the panicle on the plant, about 30 days before the reproductive phase starts. That is, the panicle start developing within the stem 30 days before. Normally before that duration is, you can say, considered as vegetative duration. So if we can know the flowering time, and also 1 or 2 plants if we check when the panicle initiation starts, then you can decide whether your plant is in the vegetative phase. So in the vegetative phase stress, there will not be any difficulties, because say in the initial seedling stage, before the panicle initiation starts, if you give, it is only vegetative stress. It is not going to affect the panicle development, or which decides the number of grains later. So that is possible. Only when we want to do reproductive stage stress, you have to adjust only your sowing time. Other than that, there is no option. Either you do sowing of your planting of the wild type earlier, or the transgenic plant later, so that the panicle initiation phase matches in both the genotype.
Okay. So, but same means same condition, like
Yeah, in the same condition. Especially if you are using drought, you should understand like remember that you give equal amount of soil moisture, and if the plant sizes are different, then you can also do phenotyping in the same pot if you want to know whether it is giving dehydration tolerance or dehydration avoidance. Normally when we do plant wild type and transgenic in separate pot, so the tolerance we see in transgenic may be because of the dehydration avoidance or dehydration tolerance, both mechanism. But if you put in the same pot, if you put transgenic as well as wild type, then mainly the difference, if it comes, it is because of the cellular tolerance mechanism, because water is available there in the pot, it is equally to both the plants. So only if there is difference in the tolerance mechanism, then you will see the difference. So you can do both the kind of experiment in the plant to see what kind of mechanism it contributes.
Okay, thanks.
Any other questions? Okay, if not, let's thank sir for his wonderful talk. Let's go for a tea break, then we come back here at 12:15 or 12:20 for starting the next session on student presentation. Okay.
So thank you all.